Dr. Nicole Wagner, who is currently the CEO and president of LambdaVision. The technology of LambdaVision, which we'll hear more about in this talk, was developed from research Nicole conducted while completing her PhD at University of Connecticut. Today, she's going to speak about the performance and stability of protein-based artificial retina that was manufactured in microgravity. So with that, Nicole.
All right, so I think you guys should be seeing my screen right now. All right, so thank you very much, Rachel, for inviting me to this presentation today. What I'm going to do is I'm going to talk a little bit about LambdaVision at first, and then I'm going to talk about some of the work that we're doing in a microgravity environment, along with our implementation partner, Space Tango. So just a little bit of history. LambdaVision was founded to develop and manufacture a protein-based artificial retina to treat patients that are blinded by end-stage retinal degenerative diseases. Some of the ones that you're most common with are probably macular degeneration, retinitis pigmentosa, and all of those diseases are devastating diseases which ultimately lead to blindness. And so our product is designed to help those patients at the end stage when the majority of their vision is lost. Let's see. So I like to start all of my presentations off asking everyone to imagine being at the height of their career and being forced to give it all up because you can't see a computer screen. Now imagine having to ask your friends and family for a car ride because you have lost your license. Finally, imagine finding out that you're about to become a grandparent for the first time, only to know that you'll never be able to read your grandchild a book. Retinitis pigmentosa affects about 1.5 million people globally. It affects about 100,000 people in the United States, and it is designated as an orphan designation by the FDA. This disease affects your peripheral vision first, and over time that will start to narrow until a patient is completely blind. Age-related macular degeneration, it affects a much larger patient population, about 30 million people globally, about 10 million people in the United States. And this disease works in the opposite way of retinitis pigmentosa, affecting your central vision first, and over time that'll eventually expand until a patient is completely blind. Now there are some treatments available for these diseases, but most of these treatments only slow the progression. So a lot of people are familiar with anti-VEGF treatments. These are these injections into the back of the eye. What you'll see here on the left-hand side of your screen is a picture of a normal person being able to see. That's actually a picture of our founder there. And on the right-hand side, you'll see the current standard of care. And you can tell that this is very low, digitalized, pixelated vision, barely enough to distinguish between light and dark. And this is just not good enough. This technology has set a reimbursement precedent. It's currently being reimbursed at over $75,000 per eye for just the device alone. And so this is just not good enough resolution for the device. So what LambdaVision solution is we have a high resolution artificial retina that can be placed into the back of the eye through a simple surgery, something similar to a retinal detachment procedure, which is really important because it increases adoption potential by retinal surgeons. Additionally, the implant is small, flexible, and is long-lasting, which is really important. And our implant is intended to replace the function of the damaged rods and cones of the eyes, which is your light-sensing cells of the eye. So the implant itself makes use of the light-activated protein Bacteriorhodopsin. Bacteriorhodopsin is a light-activated proton pump, where it will pump a proton from the intracellular surface of the protein to the extracellular surface of the protein. And it does that because it has an all-trans retinal chromophore, which is covalently linked to a lysine residue in the binding pocket. The protein itself also has incredible stability because it's arranged in this two-dimensional hexagonal lattice of trimers. So this gives the protein a stability which exceeds over 85 degrees Celsius, making it a really great candidate for device applications. It's that construct that you'll see on the bottom left that is integrated into our retinal implant construct. So we actually use an ion permeable scaffold, a layer of a polycation, and then multiple layers of protein, polycation, polycation, so that we can absorb a sufficient amount of light to generate an ion gradient that can stimulate the remaining neural circuitry of the retina. So what's happening in your eye? I hope everybody who's watching this presentation has a nice healthy retina, and you're probably sitting in your office right now, and what's happening is the light from your office or wherever you are is actually entering your eye, and it's being converted into a signal that's being sent to the bipolar cells, the ganglion cells, and eventually to the optic nerve in the brain. People who have retinitis pigmentosa or macular degeneration, they lose those light sensing cells, or your photoreceptor cells. These are your rods and cones. As those cells degenerate, your eyes become insensitive to light. So they can't capture light and convert it into a signal that can be sent to the brain. So what we do is we put our implant where those cells would normally exist. Now the protein will absorb light. In response to light, the protein is going to pump protons towards the bipolar and ganglion cells, which are picked up on receptors, and that's eventually what is going to send a signal to the optic nerve. So there are some competitors out there that are developing artificial retinas and retinal prosthetics, but what you'll really notice here is that these are really engineering marvels. You have battery packs, wires, goggles, and with that added complexity comes a much more complex surgery, which is certainly a challenge for people who are going to be putting this into the back of the eye, and it increases the risk. Additionally, because these are electrically stimulated, you need external hardware. Because we are using a light activated protein, we don't have to have any external hardware. And then most importantly, we're not limited by the number of electrodes on a chip. Rather, we're looking at molecular packing of protein molecules onto the scaffold, which allows us to get a much greater pixel density than the competing technologies. So to give you a sense of what that pixel density looks like, again, this is the first figure you saw where we see Bob Burge on the left. This is the current standard of care, which is about 60 pixels, or about 3 pixels per square millimeter. And now this is what you can see with LambdaVision's artificial retina. Now you can make out edge sensitivity, facial features. All of the people who would receive this will see in black and white, and that's primarily because your light sensing cells are responsible for the color vision. And those are the cells that are damaged in these diseases. Let's see. So there also are a number of emerging technologies that are out there. I'm sure many of you are familiar or have heard of gene therapy, optogenetics, stem cells. You know, a challenge with a lot of these technologies is the ability to get a high enough number of, or optical density of the protein rather, to be able to generate a signal that can actually get a sufficient ion gradient to stimulate those cells. And so this image here is a picture of a rat with a fiber optic in their brain. And that rat has that fiber optic because that's the light that's actually stimulating those cells. Additionally, I didn't mention this in the beginning, but retinitis pigmentosa is controlled by over 100 different genes. So to make a gene therapy, it's very costly and very expensive to do that. And it only targets a very, very, very small patient population. Our implant is intended to help the patient regardless of the gene type, which is a huge advantage there. And then, you know, there's a lot of work being done in stem cells. I think stem cells are great for trying to rescue areas of the retina, but in terms of actually recreating that neural circuitry, it's very, very challenging. So how do we make these retinal implant constructs, and how does that relate to our project with ISS National Labs and NASA? So the way that we actually manufacture the artificial retinas is through a layer by layer process where we treat this ion permeable scaffold, and then we do a series of dipping and washing steps to generate a multi-layer thin film. And you can see that on the left-hand side here, where we have that ion permeable scaffold, a polycation, Bacteriorhodopsin, polycation, Bacteriorhodopsin, and we do this over 100 times. And then you can see in the right-hand figure that it is layer dependent. So the number of layers that you add, you can actually absorb a greater amount of light and generate a higher signal. And so what we're really looking for is a consistently oriented thin film that will be able to generate a unidirectional ion gradient. The way that we're currently manufacturing these, we actually manufacture these in larger sheets, then we punch them, package them, and then terminally sterilize them. So there are, some limitations to how we do this on Earth. And what you'll see here is a picture. Actually, these are taken from our lab. So the way that we're doing this, we are subject to the effects of gravity. We're subject to things like evaporation, sedimentation of solutions, and all of that ultimately translates to a challenge in terms of implant homogeneity. And so any issue that we may have in layer 1 can be compounded as we build up these layers, which overall reduces the homogeneity and ultimately stability of those thin films. And then, what it also causes the challenges in terms of the quality assurance and the assays that are needed to look at the quality of those thin films. And that can ultimately reduce our usable area for preclinical experiments and ultimately clinical trials. So how did we get started in space? It's a question I'm asked a lot. And, if you would have asked me, 4 or 5 years ago, if we would have been looking at manufacturing this on the International Space Station, I probably would have said no. And it's not because I couldn't conceive of it. It's because I just didn't know how to get there. What we did know is that we had a lot of challenges with this manufacturing. And, we were in sort of the right place at the right time. We were part of MassChallenge in 2016. Where we received, at that time, it was the CASIS Boeing Sidecar Prize. What was great is that at that time, we were paired with our implementation partner, Space Tango, which really helped us to see, we knew what the challenges that we had on Earth with our implant were. What we didn't know was how to do that in space. And that's really where Space Tango came along, and as well as working closely with the ISS National Labs. So following 2016, we spent about a year thinking about and designing the experiment that we were going to fly to the ISS. We actually flew our experiment on Space X 16 in December 2018. And then the experiment returned in January. We evaluated the protein in the thin films then. So there's 3 pictures here of what our cube lab looked like, the inside of the box, and then on the far right, you can actually see the protein flowing over the thin film. What our hypothesis here is that we would get a much more homogeneous, stable thin film that would increase the performance of the thin film. We also know, in terms of looking at this as just a design and experiment, the protein is incredibly stable. So it allowed for us to be able to send the protein to space and not really have to worry about any degradation. This whole process, again, was very efficient because it was automated. So we didn't, we were limited in how much astronaut time that we needed, limited in the amount of resources that we needed from ISS. This was all very, very compact. And also, in terms of design, these implants are very, very small. So we can make quite a few thin films in, a 2 to 3 cube lab, which makes this also a very, cost efficient, as well as feasible in terms of the production volumes required for our clinical trials. And, just kind of touching on that again, as I mentioned, our target is going to be the orphan indication of retinitis pigmentosa first. Because we are targeting an orphan indication, we're going to have a very small clinical trial. So this is a really great use case for us to be able to do a very manageable production volume as well. So this figure here is really just to talk a little bit about the protein stability. I mean, one of the things that almost would have been it makes it a non-starter is if the protein really changed a lot in a microgravity environment. And what this figure here is showing is that pre-flight and post-flight protein maintain functional integrity, which was a really, really important thing for us, and a really great outcome from that very first Space X 16 Pathfinder flight. So some of the important learnings that we got from that Space X 16 flight, layering was successfully demonstrated in microgravity. That was really important. We were able to deposit 100 layers onto certain areas of the substrate. I think in that very first image, what you saw are some bubbles. One of the things that we are doing now for our second flight is trying to reduce that bubble formation. And we've been working very closely with Space Tango and some people looking at fluid dynamics to change the design to remove those bubbles. We didn't see any double layering, which was really, really important as well, because we did not want to have coating on both sides of the thin film. And one thing we finally observed is that there was some cross-contamination of the solutions, but we have been working very diligently to solve that over the past couple of months. And we also were awarded a phase 1 SBIR, which helped to look at optimizing some of those parameters, and that phase 1 is listed below. So very recently, we were awarded a NASA NRA award along with our implementation partner, Space Tango. The goals of this award are really to explore the benefits of microgravity for the development of our artificial retina. What we also want to do is evaluate and improve on-orbit manufacturing processes to produce artificial retinas that will be examined then on Earth. And then, I think what most people are excited about too is to establish good manufacturing practices and capabilities that are going to be necessary to produce any product that could be manufactured in microgravity that could have clinical benefit, which is critical. So as we think about this, what's going to be really important is that GMP piece. Our goal is going to obviously to manufacture an implant that's going to be safe and effective. As we're looking at establishing these GMP capabilities and working alongside with Space Tango, things such as reproducibility, consistency of manufacturing, as well as looking at that stability of the protein, as well as the stability of the artificial retinas, it's going to be critical moving forward. And then alongside that, some of the other considerations here are making sure that we have the appropriate assays, characterization, and documentation that are going to be needed. And so we have, we're at the very early stages of doing that right now, and are eager and on track to move forward to having those appropriate discussions with the FDA and NASA and the ISS, if and when it's necessary. So this is my final slide. I think one of the really important pieces is beyond, the artificial retina technology that LambdaVision is focused on, are really some of the other technologies that can really come from layer by layer manufacturing. Just looking at the protein Bacteriorhodopsin itself, I mentioned its stability, as well as some of its light activated properties. It can also be used for photovoltaics. So one of the things that we're doing with our NASA Phase 2 SBIR award, which was recently awarded to us, is looking at some of these other technologies, both for Bacteriorhodopsin, applying it on different scaffolds. And I think, long term, you'll be able to see some additional opportunities coming out of this, especially as we start to establish those processes and procedures and quality control systems that are necessary. And, I can see the future being things like drug delivery, biosensors, wound healing, and tissue engineering as well. So with that, I thank you guys for your time.